Alrighty. Hi, everyone. I hope everyone's here for the Biochem review, Biochem 1 for 4053. Yes? All right, cool. So I have these surveys going around, and they're just evaluations for the SI and the review itself. So all I need to do is, on this top corner, we're going to write 5-5 as our section.
Shane, do you have a mic?
Yeah.
Can you use it?
The thing is, like, the mic is for online streaming. So I have to speak up to everyone, but the mic is for those people to hear me. So welcome everyone that's online as well. So I'm just going to pass these out. Just remember 5-5 is the section. Thank you. 5-5, and then on the top.
This one works, but it's really, really sensitive. But this is for the mic in here.
Okay, cool.
So try it.
Hello, check 1, check 1, 2, check 1, 2.
Okay, so we can put it up closer to you
Okay.
if you want. You can adjust it. But this one works for the mic in here.
Okay, awesome. Thank you. Hello, hello. Check 1, 2. Okay, can you hear me? Can you hear me? How about now? Is this better? No? Yes? You can hear me? Through the mic? Okay, cool. All right, so anyone that isn't in Dr. Hashim's class, can you raise your hand? Or just, is there anyone that's not in Dr. Hashim's class? Because I just wanted to show, if you're not from the class, that the way to get to my review is, so my name's Shane, and under the review, you see Biochemistry, Shane S, and you just click on that document. So if you need to pull up the review, that's going to bring you to a Word document, and I have a lot of multiple choice questions that we're going to try and go over in the allotted amount of time. Okay, cool. All right, so to start the review, I want to mention a disclaimer. So this review is not everything that's going to be covered for the final, and I've taken things that I find most relevant to the course, and I've kind of made up multiple choice questions. And these questions are also from, presented from Oxford Press on biochemistry, so you can Google these, and they're all online. They're just specific questions I thought were the most prevalent to the material that we've covered this semester. And they've also been a little clarified with the professor as well. So to begin, I'm going to read the question, and then we'll take about a minute for everyone to solve it, and then we'll kind of go over the answer. Sound good? Okay, cool. So number 1. So it says, when a mixture of glucose 6-phosphate and fructose 6-phosphate is incubated with phosphohexodase isomerase, the equilibrium mixture contains twice as much glucose 6-phosphate as fructose 6-phosphate. Calculate the delta G standard at 25 degrees Celsius and given the constant. So we'll take a minute and see if we can solve that, and then we'll go over. I still see some people writing, so I'll give a little more time. All right, so first question is, what is the formula for delta G naught, or under standard conditions? Does anyone know? Awesome. So delta H equals, or sorry, T delta S. Awesome. Equals delta G naught. So now we're plugging and chugging. So for delta H, what is our delta H in this particular equation? Is there a delta H? So can we use this formula to solve for delta G naught? So is there another formula that incorporates the given variables? Yes?
Negative RT ln K equation.
Awesome. So delta G equals negative RT ln K. So what is R? Given. And that's our constant. What is T? Temperature. And what is temperature in this particular formula? Awesome. So I heard Kelvin, so that's awesome. How do we get Celsius to Kelvin? 273. So to get to Kelvin, we want to take the Celsius and add it to 273. So what do we get for Kelvin in this equation? 298. Awesome. So 298 Kelvin. So our last part is our K. So how do we solve, or so ln is a natural log. And how do we solve for K? Awesome. So we have a ratio of concentration. How do we know what is on top, and how do we know what's on bottom? Products over reactants? Awesome. So we all agree? So in this equation, what is our ratio? I'm sorry? 2 to 1. So we have 2 over 1. Yes? So let's identify what the products are and what the reactants are. So what's the products? Awesome. So product. And then that would leave glucose 6-phosphate as a reactant. So we said the ratio for K is equal to products over reactants, right? So how much product are we given and how much reaction are we given? 2 over 1. So we're saying there's twice as much glucose. So glucose is a reactant, right? So would it be 2 over 1 or would it be 1 over 2? 1 over 2, because we have twice the amount of glucose than fructose. Does everyone see that? Awesome. So now we solve for our K, and now we plug and chug. So we have R, which is 8.31 joules per mole Kelvin. Sorry that I'm going to interfere with this. Times temperature, 298 Kelvin, times ln, 1/2. So what do we get as our final answer when you do this calculation? I'm sorry. Positive 1.7 kilojoules per mole. Awesome. So we converted that from joules to kilojoules. And when we first got the equation, did everyone get like 1700 before? When you calculated it? So you got 17.5. So when you take that from joules to kilojoules, you divide by 1,000. Does everyone understand that? Thank you. So we understand how the ratio was produced. We saw that we plugged in Chug for our givens, and we calculated Kelvin. Does anyone have any questions on how to solve for delta G standard? No? So we're ready to go to the next equation? So the answer is C. Cool. Okay, so for the next equation, or the next problem, the problem says an endergonic reaction with a blank delta H and a blank delta S can be changed into an exergonic reaction by decreasing the temperature. So we'll take a couple minutes, or a minute to solve, and then we'll discuss. Okay. So is everyone ready to start the question? Yep. Okay, cool. So first question is endergonic reaction. What is an endergonic reaction? Positive delta G, and it needs energy. So is energy exiting the system, or is energy entering the system? Entering. So I remember ender, so En, enter. So the system is taking heat. Exergonic, heat is exiting the system. And you said a positive delta G. So for our endergonic reaction, we have a positive delta G. Awesome. So based on a positive delta G, what would our delta H be and our delta S be? Or before we go into that question, what's the formula for solving for delta G, given the variables? Awesome. So delta G equals delta H minus T, or multiplied by T minus S. So based on that equation, if we substituted, what could we substitute in that would allow temperature to be the dependent factor from changing a positive delta G to a negative delta G? So let's just plug and chug and see what happens, because that's usually a good way to solve. So if we have a delta H that's a positive and a delta S that's a positive, so that's the first choice, that would leave us with what? So temperature is getting more negative. So we have positive minus a negative times a positive. That would leave us with a positive number. Would that be getting us more endergonic or more exergonic? More endergonic. So we can eliminate choice A. So again, let's plug and chug. So delta G, we're giving a negative value minus a negative value minus a negative value. So for this equation, what would our delta G be getting? More endergonic or exergonic? Exergonic? Awesome. So we can leave this as a choice, right? So again, if we substitute positive as well as positive minus minus. What would that leave us with? Depends. So it depends on the variable. So what's a more important factor, delta S or delta H, in determining exergonic or endergonic? What's more variable? H usually is steady in the reaction. Heat is either in the system or it's not in the system. It doesn't really fluctuate. But S can be manipulated depending on the hydrophobic effect and how much is already contained energy inside of it. So if it's highly disordered, so that's usually the variable in this equation. So when we're getting a negative number for S, we can correlate to say that could be that could create an exergonic reaction, but most likely it's going to be affecting, the positive will have an effect on it. So let's try the next one. So we have 1, 2, 3, 4. We have delta G equaling a negative minus a negative plus a positive. So then S would be also contributing to being more positive. So then it's still affecting by this negative value. And if none of the above, probably is not the answer. So we can eliminate none of the above because that wouldn't make sense. So out of all the equations, which one is not contradicting? Is there not a contradiction in where we're having a clear statement that says delta S is going to be, or delta G is going to be negative? B? Does anyone have any questions why it wouldn't be B and it could be C or D? No? Everyone's good with B? Okay, cool. Okay, so everyone can see that one. So for the third question, how does the hydrophobic effect influence the structures of large molecules? A, nonpolar molecules are not easily soluble in water and aggregate. B, polar groups are oriented on the surface and interact with water. C, nonpolar molecules can mask the polar characteristics of the hydrophilic molecules. D, A and B, E, A and C. What do we think? Okay, so A, why? Awesome. So we have micelles and we have these aggregation forming with nonpolar structures. And we can call that a hydrophobic effect. Awesome. What else do we think? Okay, awesome. So why do you think B is right? Awesome. So we have this, we want to be stable, this most stable structure we can. And this goes back to like the micelles as an example, how those inner molecules are going to be nonpolar and those outer molecules are going to be polar, because most of our solutions are going to be aqueous. And they're going to have H2O. So what about C? Do we think C is correct? No? Okay. So why don't we think C? Awesome. Because do we really have polar characteristics in non-polar molecules? So that doesn't make any sense. So our answer is D. Any questions? Okay, let's move on. Okay, everyone can see that? Awesome. So 4, you mix equal volumes of 0.05 moles of NaH2PO4 and 0.05 moles of Na2HPO4 and the pKa's for this phosphoric acid are 2.5, 6.82, and 12.38. Which of the following best describes the resulting solution? Does anyone think they have an answer? B? Why do you think B?
Every answer besides E would be absurd. It probably is buffered because it has equal concentration of conjugate base.
Awesome. So a buffer really doesn't want to vary in that acid and base concentration. So we really don't want something really acidic, and we don't usually want something really basic. Any other explanations why B would be the correct answer? So how do you determine if something is well buffered or not well buffered?
Usually look at the pK, right?
Yeah.
And if they're equal concentrations, then pH will equal pKa.
Awesome. So when pH equals pKa, do we have a term for that? Optimum buffer capacity. So we can use titration curves to understand how to figure out what the optimal buffer capacity is. Through a titration curve, what else can we find out? Isoelectric point, so Pi. So Pi, what is that really saying about the molecule? Net charge is 0, so awesome. So it's in a stable, that neutrality is going to give that molecule the less ability to react. So in this molecule, we have 3 pKa's, and we could find Pi for this molecule. And based on that, we would want to find the pH that's closest to that Pi. Does that make sense? Do we have any questions? No? We're okay to move on? Okay, cool. So the Henderson-Hasselbalch equation, it allows for graphic determination of molecular weight of a weak acid from its pH alone, does not explain the behavior of di- and tribasic weak acids, employs the same value for pKa's for all weak acids, is equally useful with solutions of acetic acid and hydrochloric acid. E relates the pH of solution to the pK and the concentration of acid and conjugate base. Does anyone think they have an answer? E? E? For Einstein? Awesome. So what's the equation for Henderson-Hasselbalch? So HA over acid? Or is it the other way around? Other way around? So conjugate acid over base. Oh. Does everyone agree? Cool. Awesome. So that's E. So for number 6, the pKa1, pKa2, and pKr for the R group for the amino acid arginine are 1.8, 9, and 12.5 respectively. At pH 7, arginine would be charged predominantly as follows. So I'll give 30 seconds to figure out this one, and we'll go over what we think the answer is. Okay, so first strategy I would use when tackling this question is to draw out what arginine looks like. So does everyone know what arginine looks like? Yes? So we have a backbone, NH3 or NH2. Minus one carbon. So for our alpha carboxylate, which side is the carboxylate on? That's not arginine? CH, 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 NH. So there's an NH before the NH2. My bad. Okay, hold up. So awesome. Thank you. So for our alpha carboxylate, where are we finding that on the molecule itself? C terminus? So what about our amino? N terminus? Awesome. And our guanidino? R group. So we can all, this is our R group here. So respectively, how do we distinguish pKa's? Which one do we give 1.8 to? Awesome. So pKa for our carboxylate is 1.8. What about 9.0? Amino? So for most amino acids in their structure, your C terminus is going to be around 1.8 to 2.3, and then our N terminus is usually going to be 8.5 to about 9. So then that leaves us with our pK of 12.5 for our R group. So based on that, at a 7.0 pH, what would be our alpha carboxylate charge? Negative. So we're going to have a negative charge here. Awesome. So that leaves us with B and D, or B and E. What about our alpha amino? Positive. Awesome. So that leaves us with B. And we'd have a positive charge where on the guanidino group? NH2 plus. So would that be with the double bond or the single bond? Awesome. So the overall charge of this molecule would be what? +1? Awesome. So overall charge. Does anyone have any questions on how to determine charge based on pH? So a good, let's define when something is going to be charged or not charged. So if pH is greater than pK, what does that equal to? So for the example, pH is greater than 1.8 for this carboxyl group. Does that mean that it has a charge or it doesn't have a charge? It has a charge. So if it was lower than its pKa, what would happen to it? It wouldn't have a charge. So what would bind to it? A proton. So it would be protonated. So usually, depending on what the charge for that given species is. So say it's a positive molecule, like arginine. But we could have a negative molecule like, what's a negative molecule, for example? I'm sorry? Glutamic acid. So glutamic acid, what's its pKa? 2? Does anyone know? So let's say its pKa is 3, and you have a pH of 2. So its pH is lower than its pKa. What's that glutamic acid charge going to be? Positive? Even though the molecule, when it's deprotonated, is negative? It's usually going to be neutral. So if a molecule is, when it's deprotonated, is negative, if it binds that protonation, it usually becomes neutral. So it doesn't give a charge. Does that make sense? Does that make sense? Okay, cool. So those molecules range depending on its pH. Does that make sense? Does anyone have any questions? We can practice more at the end, and we can go over that if we have any more questions. Okay, cool. So for number 7, which of the following statements about the structure of nucleotides is correct? A, the sugar component of nucleotide is always ribose. The sugar component of a nucleotide is always deoxyribose. C, the bases in the nucleotides are attached to a pentose sugar motilates by a glycosidic bond. And D, the sugar component of a nucleotide is in the L configuration. Does anyone have a guess? C? So why do we think C? That's a great reason why. So what happens if, at what position could we have a ribose be deoxyribose? 2? 2? And what form of structure would deoxyribose be in? DNA? Awesome. So DNA. Could we have a sugar molecule in DNA be an L conformation or configuration? No? What usually is the configuration for DNA? D. So most sugars are in D conformation or configuration. Cool. Any questions? All right. So phosphodiester bond, what is the definition? So A, links a base to a pentose nucleotide. B, joins adjacent nucleotides in one strand. C, joins complementary nucleotides in 2 strands. D, difference between a nucleoside triphosphate. What do we think? B? Okay, why B? Awesome. So B is the correct answer. So joining adjacent nucleotides in one strand. So A links bases to a pentose in a nucleotide. What type of bond is that? Glycosidic. So in the previous question, we could draw out where the glycosidic bond happens. And what carbon does that usually happen on? 1,4? So I see position 1 when I drew out a molecule. So I'll show you guys. So here's an example of a N-glycosidic linkage. So that carbon 1, let's say that's this one. Well, this would be carbon 1, considering that it has the O double bond. So this would be carbon 4. And carbon 4 then would be linked to a nitrogen, so we'd give it N-glycosidic linkage. So where on this do we see a phosphodiester bond? Carbon 2? Awesome. So carbon 2, we have that OH, this hydrogen wouldn't be here, and that attaches to the next ribose on the bottom. Does that make sense? And that would be position 5. Sorry, you're right, because this technically has another carbon before it bonds to this one. So this would be carbon 1. This would be 2. This would be 3. This would be 4. And this would be 5. Does that make sense? So the attachment is at carbon 3 to carbon 5, or the phosphate group, and that's our phosphodiester linkage. And our glycosidic bond occurs at carbon 1. Does anyone have any questions? Okay, cool. So for the next question, number 9, reactions by which chemical energy that has been stored in a high energy phosphoanhydrate bond in ATP is released. What is the process called? Hydrolysis? Awesome. So ATP phosphorylation, what does that? Awesome. So what adds phosphates? What type of molecule? Kinases? Awesome. How about ATP dehydrogenases? Or hydrogenation? What is occurring there? Dehydration? So what are we losing? So we lose water. And then for this one, hydrogenation, what are we gaining? Awesome. So 9 is D. Number 10. So Chargaff Rule state A, the amount of guanidine plus cytosine in a mammalian genomes varies from about 39 to 46%. DNA is always double-stranded and RNA is always single-stranded. DNA contains 2 grooves, a major groove and a minor groove. In DNA, the number of adenine residues is identical to the number of thymine residues, and the number of guanosine residues is identical to the number of cytosine residues. E, DNA contains 2 strands and run in opposite directions. D? Awesome. So that's just straightforward. That's his rule. Any questions? Okay. Number 11, transfer RNA molecules are involved in translation, transcription, replication, reverse transcription, post-translational modifications or processing. Translation, awesome. So what type of molecule is used for transcription? DNA polymerase? RNA polymerase? RNA polymerase, awesome. What else? What's the type of strand that's used? Yeah, mRNA, awesome. So mRNA is used for transcription. Translation, we have tRNA and the processing of mRNA. Replication occurs with DNA. And reverse transcription it causes, which is used in HIV, with people that have the virus, it retrotranscribes itself into our DNA. So post-translational modifications, where do they usually occur? I'm sorry?
So the Golgi apparatus.
Golgi? What were you saying?
That's usually just like when they take out the introns.
Take out introns? Anything else?
Well, isn't taking out introns before translation?
So post-translational modification can occur, yes, mRNA will be cut down, so that'd be post-transcriptional modification, but you could have introns that are still left over that are still taken out before then. I mean, after then, depending on further transcription could be processed, depending if you're making mRNA or DNA.
Okay, so translation's after the protein is made, right?
Mm-hmm.
So we're not working with mRNA anymore.
Correct.
Because I think wouldn't introns be out by now because introns are taken out before it leaves the nucleus, right?
Correct. So introns would be gone before translation.
Yeah, so it would be like pre-translation.
Okay. So introns, so mostly is occurring in the Golgi, and we have, what would be processing post-translational modification? What type of enzyme? Glycosylates? Ribosomes? So ribosomes is usually that structure that's going to really be able to tell if that tRNA is able to recognize its codon and anticodon. So it's kind of that area where these are what's controlling the process. Cool. Any questions on 11? So answer was B. So for 12, which amino acid does not have a primary alpha amino group? Proline? Awesome. So what does proline's structure look like? Cyclic. Awesome. How many carbons are in proline? 5? Awesome. Does everyone understand alpha amino group? Okay, cool. So, any questions? Okay, number 13. In Watson-Crick's model of DNA structure, A purine in one strand allows hydrogen bonds with purine in another strand. B, the 5 end of both strands are at one end of the helix. C, the bases occupy the interior of the helix. D, A, T pairs share 3 hydrogen bonds. E, all of the above. Oh, sorry. C? Yes? C is the answer. Why? Why wouldn't 2 bonds? Awesome. What relationship has 3 bonds? C and G. And what does that mean about C and G bonds? Higher dissociation. Awesome. So it's a stronger bond, because more hydrogen bonds, the more likely you're going to be less likely to be broken. Awesome. So it has 3. So was Watson-Crick's model explaining 5 ends on both ends of the strand? It's 5 to 3. And a purine in one strand always allows hydrogen bondings with a purine in another strand. Is that true? No? What is a purine? What are the 2 types of purines? Adenine and guanine. Awesome. And what are our pyrimidines? Sweet. Any questions? No? Okay, moving on. So 14, we will sodium didecyl sulfate polyacrylamide gel electrophoresis, or SDS page, provides important information on A, electrophoretic characteristics of a protein, B, net charge on a protein, C, total molecular weight of a multimeric protein, D, molecular weight of a protein subunit, E, affinity of a protein for acrylamide. D. D is the correct answer. Why do we think D? Or let's ask a better question. Why is it not C? Because that would be, for me, the alternate choice if I had to choose. Why isn't it C? Awesome. So we have restriction enzymes.
Well, SDS page uses protein, right? You know, restriction enzyme for protein. It's because you use heat and beta-mercaptan ethanol to break the multivariate mechanism.
Awesome. So we can add 2,3 beta mercaptan, or is it two?
That's 2 mercaptan.
Yeah, it doesn't matter. Mercaptan ethanol, honestly. You know mercaptan ethanol, you're good. So those break, what did two captain ethanol break? What type of bonds? Disulfides. And what are most proteins structures held by? Disulfide bonds. So breaking those apart, we would have, what would happen to if we had a multimeric protein? Dissociate into its smaller protein subunits. We also have heat. So what does heat do to proteins? Denatures, so we could have proteins denaturing, causing them to further break down their multimeric subunits. So that's why we can eliminate C. And what type of assay or chromatography do we use to find charge? Ion exchange chromatography. Do we all agree? Okay, so for charge, we can use ion exchange. For affinity, what type of proteins or type of chromatography can we use? Affinity chromatography. Pretty straightforward. At least one thing is straightforward in biochem. Affinity chromatography. So what could we use to tell affinity chromatography? What type of substances could you have a specificity for? So first thing that comes to my mind is antigens and antibodies. So you can specifically put in a column antibodies that are specific to an antigen. So that's a type of affinity chromatography. Awesome. Any questions? No? Okay. So number 15. While proteins are usually composed of linear chains of amino acid, branch chains of amino acids and internally cross-linked chains can be found in a certain proteins. Polypeptide chains are most commonly linked to each other through disulfide bonds. So that kind of correlates with the previous question. Any questions on that? Cysteine.
That's where the disulfide bridge usually form between cysteine molecules.
Yes. And it depends on what we're talking about. So if we're talking about alpha keratin, or if we're talking about a specific molecule, those will arrange differently. But cysteine is the most common. Does everyone agree? Yes? So we have cis. It's not how you spell cysteine. I'm horrible. All right. 16. Which of the following can be extended by DNA polymerase? A, a single strand of DNA ending in a nucleotide with a free 3 hydroxyl group. B, an RNA strand with a ribonucleotide with a free 3 oxygen or hydroxyl group. C, a single strand DNA in a deoxyribonucleotide with a free 5 phosphate group. D, a single RNA strand. E, all of the above. E? A? A is the correct answer. So a single strand of DNA ending in a nucleotide with a free 3 hydroxyl group. So what way is DNA read? 5 to 3 primes. So where can free nucleotides be added to? 3 prime. So on that single strand of DNA, we always have to have a 3 prime hydroxyl group in order for a new nucleotide to be added. And the new nucleotide is going to be attached to what part on the nucleotide? The ribose sugar? And what's going to be on that ribose sugar? Phosphate? Awesome. So that phosphate is going to bond to that 3 group on the ribose, and then a new 3 hydroxyl group is going to be allowed to bond to the next one. Does everyone kind of picture that? Does anyone kind of want to see a picture? No? Everyone's okay? Okay, cool. So can no, never mind. Okay. 17. Which group is attached to the pyrimidine ring in thiamine is not usually present in uracil? C? Awesome. C is the correct answer. So what is another name for thiamine? 5-methyl-uracil. Awesome. So another name is 5-methyl-uracil. So if you look at the molecule of C-uracil, the only difference is at position 5, we have a methyl group. Any questions? No? Cool. Moving on. Number 18. Inside our cells, free nucleotides are almost always associated with C? That is correct. So magnesium, what does magnesium do to the ability to grow a DNA strand? Stabilizes? Awesome. So we stabilize, how many magnesium molecules are there in order to stabilize the DNA strand? 2? Awesome. And what are they attached to? Awesome. So that phosphate molecule is going to be able to be coordinated inside the magnesium. So the charge of magnesium is positive, and our phosphate group is negative. So that's why we have a counter ion. And that's why you need magnesium in your diet, or else you couldn't synthesize new DNA. So there's probably a lot of correlation with a lot of cancers or anything that's mutating in our body with maybe improper functioning of DNA stabilization. So magnesium supplements could possibly be increased. So 19. Any questions on that previous? No? Okay, 19. Which of the following nucleic acids provide the key to decoding genetic information? A, RNA, rRNA, B, mRNA, C, tRNA, or D, DNA, E, none of the above? A? Why would it be A? Okay, that's a good guess. Does anyone else have any other choices? DNA? Why would it be DNA? So the question is, which of the following nucleic acids provides the key to decoding genetic information? RNA? rRNA makes the template strand? That's a good guess. What do we think? C? So why do we think C? Awesome. So where does our new amino acid come on? Like where does that attach to? tRNA. So tRNA recognizes anticodons that are attached to amino acids. So in order for we to actually have genetic information decoded, we need that recognition site. So for every codon, there's that anticodon. So our tRNA molecules are really the deciding factor on what amino acid is built. So that's why we say that tRNA, which is C, is the correct answer. Does anyone have any questions? Okay, awesome. So for number 20, which of the following DNA sequences is considered palindromic? A, GAAC, TT. We'll give you a second to figure it out because I'm not going to read all these amino acids. B? Do we all agree? Awesome. B is the correct answer. So just to draw it out, we have A, G, C, T. Now, what does palindromic mean? Same forward and backwards. Awesome. So if we correlate our Chargaff's rule, for every T molecule, we have an A molecule. And every C molecule, we have a G molecule. So looking at this from a perspective, if we said this was 5 to 3, and this was 5 to 3, if we flipped that 5 to 3 molecule, we'd have the same going forwards and backwards. Any questions? Awesome. 21. Double-stranded DNA molecules can be cleaved at a specific recognition sites by A, RNA polymerase, D, DNA ligase, C, reverse transcriptase, D, type 2 restriction endonucleases, E, DNA polymerases. What do we think? I hear B, D, D. Awesome, D is correct. So let's go through RNA polymerase. What does RNA polymerase do? Synthesizes RNAs, so makes RNA. What does DNA ligase do? So it joins DNA fragments. Which specific DNA fragments are joined? Okazaki fragments. Awesome. I'm just going to write, I don't know how to spell Okazaki, so connect DNA fragments. Reverse transcriptase, again, is usually an HIV. So it's one of those mechanisms where the virus is using its genome to plug into our genome. Type 2 restriction enzyme nucleases and DNA polymerase, what does that do? Makes DNA. Awesome. So differentiating between type 1 and type 2 restriction endonucleases, what do we think? Awesome. So for type 2, we have a specific recognition site. And type 1, we either down further from that recognition site or upstream of that recognition site. So this goes into blunt ends and sticky ends. So can we say that type 2 restriction enzymes are only going to make blunt? Or are they going to make only sticky? Or let me say this. So when it's cut like this, or if it's cut like this, which one is which? So this is sticky. And this one is, does everyone agree? Yes? Anyone question?
So which
So type 2, which one would type 2 do? Blunt or sticky ends? Or can we say that they do either one? It could be either one? What do we think? That's true because, so say if this was all a recognition site, right? That area is still in the recognition site. So it can connect both, if say this was all recognition site, either way it's in that recognition site. So type 2 can make blunt or sticky ends. I just wanted to go over what's the difference between the two because that could be a possible question. Any questions? No? All right, moving forward. In molecular cloning, transformed organisms must be identified. One common method for accomplishing this involves the inclusion of blank in the plasmid. D? Yes? That is correct. Any questions? No? Okay, moving forward. 23, DNA polymerase from Thermos aquatus is used in PCR because A, it is soluble protein, B, it is stable at high temperatures, C, its genes are readily distinguished from those of normal organisms, D, the enzyme is readily deactivated by heat, effectively halting the reaction, E, it is not infectious. B? B? Yes? So why would we want a DNA polymerase to be resistant to high temperatures? I'm sorry, can you speak a little bit? Awesome. So why would we want heat first in PCR?
You need to denature the strands.
To denature those strands. So what's the overall process of PCR? What do we want? More DNA. We want to take one strand and make it into 1,000 strands and 100,000 strands. So this is kind of like those crime scenes where they find like the littlest amount of DNA. So just be careful, you're going to commit crimes. They got PCR against you. Just joking, don't commit crimes, it's horrible. So we want a DNA polymerase, and they found these in like Yellowstone National Parks in these hydrothermal vents deep in the sea where they look at these organisms that are able to survive high temperatures, and they use that same DNA polymerase and are able then to amplify more DNA. So I really think that's pretty cool that we just like happen to find life in super hot places. Any questions? Cool. Moving on. So can everyone see that? Yes. Okay. So which of the following would represent a forward primer for the 120 base pair fragment of human gene that code protein hemoglobin as shown? What do we think? B? I see a lot of people looking at me like they're confused. B? Why is it B? Good. Awesome. So the complementary strand, could we use that as its primer? No. Why not? Awesome. So identifying the original sequence can allow for initially of DNA polymerase to bind. So once DNA polymerase has something to bind to, it can start formulating a new strand, the new template strand. Does anyone have any questions? Yeah. Backwards primer? So in that case, that's a great question. So if we're looking here, and you're saying, so say there was 2 strands of DNA, and you wanted from the 3' to the 5', or the 5' to the 3', but in the opposite strand. Does that make sense? So what you would do is, this would say, let's say we took this molecule and this was the other strand now. So usually it's 5 to 3, but let's say this was 5 on this side. Then we would say this is our primer up to this point. Does that make sense? So on the opposite strand, we still have to go 5 to 3'. So if she asked for 2 primers, you could figure out based on where the 5' end was and where the 3' end was. Correct. So say this is, I was saying this was the opposite strand. If you were saying it had gone the other way around. Does that make sense? Any questions? No? Okay. So 25. Which of the following amino acids would be first to elute at a pH of 8 from an anion exchange column? And our choices are lysine, alanine, glutamic acid, aspartagine, and glycine. Lysine? Yes? Awesome. So first question that I want to ask is, when we talk about anion exchange, what is being held in the column? Positively charged is being held, so that's the thing that's going to hold on to.
You mean what's directed?
Not what the matrix is made of. What is the matrix holding? Negatively charged. So when I think anion, anytime you're saying anion or cation exchange column, I always think anion held. So whatever that is, that's what's holding. So the matrix is the opposite. Does anyone have any questions? Does that make sense? So anion molecules, negative molecules are being held. Negative. And if any cat lovers are out there, I always say cats are positives. So I don't know, I'm a dog lover. I just that's how I remember it. So anions are being held. So the matrix is positive. So based on that, so for lysine, what is lysine's charge when it is charged? Positive? What about alanine? Neutral, so I put 0. Glutamic acid, negative, awesome. Aspartagine, neutral, depending on the pH of the column. And glycine, neutral. So based on that, what amino acid? Lysine. So if I asked in a cation exchange, what molecule would elute first? Glutamic acid. Why? The matrix is negative. So they repel each other and it will elute first. Why don't the neutral molecules fall through first? Awesome. So that force is being produced to push down further than just a negative gravity. Does that make sense? Okay. Any questions? All right, moving on. Okay, 26. Which of the following pentapeptides will have the highest absorbance at 280? So first question is, what amino acids absorb at 260? Phenylalanine. Awesome. What about 280? I'm sorry? Tryptophan. Awesome. And tyrosine. So based on our choices, which of these molecules have the most tryptophan or tyrosine? D. And this was actually given to you, so you're welcome. Just joking. Just joking. Playing. Does that make sense to everyone? Which choice would be highest for 260? Awesome. Yes. Does everyone agree? So this would be highest for 260. Sweet. Moving on. Which of the following is commonly used to cleave disulfide bonds in polypeptides? A, 2-mercaptan-ethanol. B, dinesyl chloride. I don't know. C, adenoacetate. D, sodium didecyl sulfate. E, trypsin. A. So this goes back to some of the stuff we covered previously. Anyone have any questions? What does dinesyl chloride do? I'm sorry? Recognize N-terminus? What I have, it's able to make blue or green fluorescent on the protein. So you can add a specific marker to a protein and we can distinguish what's affecting it. Does anyone have any other notes about dinesyl chloride? It determines the number of subunits. N-terminal analysis. How about iodine acetate? Prevents the bonds from forming? Awesome. And I said this works with cytosine. And sodium didecyl sulfate denatures proteins, gives them a negative charge. Do we all agree? Awesome. It denatures proteins, and what was the other one?
I think it's a negative charge.
Negative charge, yes. So that's all I have that I thought was relevant for test one. This is not everything that needs to be covered for test one for sure, but it's an overview. Any questions on test one material? What time is it? 3:45, so it's about 45. I'm trying to leave the last 15 minutes for questions. Are we ready to continue? Okay, awesome. So for test two material, let's start with, for the reaction, the steady state assumption is E. E. We all agree? E is correct. So what is the value for K minus 1? What does it correlate to? What dissociation? What is it greater than? I have K minus 1 is greater than K2. What does Ks equal to? I have Ks equals to enzyme concentration times substrate concentration over enzyme substrate concentration. And then I have S concentration equals to S naught. And E is less than the product of substrate and enzyme. And that means you have a lot more substrate than you do enzyme. So you usually reach that point where saturation is at 100%, more than enzymes being able to fulfill that need in steady state assumption. Any questions? Okay, moving on. So for 29, find the initial velocity for an enzymatic reaction when Vmax is 6.5 times 10 to the power of negative 5 moles per second squared. And the substrate concentration is 3 times 10 to the power of negative 3 moles. Km is equal to 4.5 times 10 to the power of negative 3 moles. And the enzyme molarity. And the enzyme's concentration at time 0 is 1.5 times 10 to the negative 2 micromolarity. When you have the answer, just let me know. B? Awesome. How did you calculate for B? Awesome, so plug and jug. Did everyone get that? Anyone have any questions? Awesome. Moving on. So a compound that distorts the active site, rendering the enzyme catalytically inactive, is called A, an uncompetitive inhibitor, B, an allosteric effector, C, an inactivator, D, a competitive inhibitor, E, none of the above. Inactivator? D, a competitive inhibitor? So I have one person saying inactivator and another person saying competitive inhibitor. A? She counted both. So A and D? C and D. So we have our choices being C and D. So let me make sure. Okay. So she counted competitive inhibitor and inactivator as something that distorts the active site. Anyone have common concern? Yes.
Just to understand the definition of competitive inhibitor, doesn't it physically attach to the active site
Yes.
and like block it for
That's a great question.
other stuff?
So if it's competing for that active site, so it binds, so nothing else can bind to it.
So I guess that's considered distorting that active site, I guess.
In the sense distorting, yes, because it's leaving it inactive.
And why wouldn't it be uncompetitive inhibitor?
So what does an uncompetitive inhibitor do? Awesome. So binding at a different spot, that causes what to happen to the enzyme? It changes a conformational change so that that substrate that wants to bind can't bind. It does, but at the same time, if she's counting one, might as well count them all. I'm just joking. No, that's a good question to ask Dr. Ishii. because if you're distorting the active site from an uncompetitive inhibitor standpoint, that should be a right answer. If that makes sense. But the question is very generalized. It's very broad. Not to say anything that it's a wrong question, because sometimes having multiple answers is a good thing. So just a question maybe to ask her, say, if I chose uncompetitive inhibitor, why wouldn't it be the answer? Because I'm not sure. Because it makes sense when you see an uncompetitive inhibitor bind, then we have active site not being able to actively bind that molecule. A competitive inhibitor binds to the active site, but it doesn't allow the substrate to bind to it. So that's the argument, is saying, is she really asking, because she counted both the questions, is she asking what's affecting the active site, what is not allowing the substrate to bind, or is she asking what's distorting the active site? So it's one of those kind of tricky questions. So just clarify maybe before email, office hours, and see what she thinks is. This is number 30. The answer I have is D. She counted non-competitive inhibitor. So she counted inactivator, competitive inhibitor, and non-competitive inhibitor. Let's see if anyone can. Yeah. Does anyone have their old test too? Was the one answer that she did put was competitive? Yeah. That's what I have too. So I have competitive inhibition as the answer.
Yeah, that's what I have. And on the answer sheet you gave us,
Yes.
you also bolded some answers. There's two, and then didn't bold the other ones.
And then for E, on the test, it's a different question? Which compound binds to the active site? So which compound binds to the active site? Competitive inhibitor. Okay, let's look at the question of the test. So 11, which of the following represents a compound that binds to the active site? Is that what we're saying? Yeah, right? So which of the following binds to the active site of a given rendering the, so which one binds? Competitive inhibitor. So this one, distorting the active site, is a little different. So that would be uncompetitive. It's weird because I have competitive inhibitor. For that question, for 30 or for 11? She has both competitive and inactivator. Okay. So for my question on my review, I have the answer as D. So if that, you guys want to clarify that and see what you think, what Dr. Sheen thinks that is, I think that'd be a good question to ask. Because it is different from question 11 in the idea that it's asking distorts versus affecting active site. Or binding to the active site. Are we all clear with that? Thank you. Cool. All right. Moving on. Do you have the time by chance? Yes. Okay. 31. So, given the following data on the rate of the reaction with and without inhibitor. Without inhibitor, we have a Km that's 0.02 millimolars. With an inhibitor, we have Km, which is 0.01 millimolars. Vmax without inhibitor, 150, and with an inhibitor, 90. What type of inhibition is consistent with these results? Uncompetitive? Do we all agree? So what is occurring? So Km, is it increasing or decreasing? Decreasing? What about Vmax? Decreasing? So based on that, what is been consistently to show with what type of inhibitor to have a decreasing Vmax and a decreasing Km? Uncompetitive? What do we think? Uncompetitive. That is correct. So let's go through. So if you have increasing Km, what is that? Competitive? So competitive, increasing Vmax. What about mixed? Oh, it's Km, sorry. Km. What about mixed? Awesome. Vmax can increase or decrease. What about Km? Decrease? Do we all agree? So Vmax can only decrease in mixed? Do we all agree? So Km can increase or decrease, and Vmax can only decrease. Awesome. That's a good question. What do we think? Could it be mixed based on the results? Yes? Does anyone disagree, and why? Why couldn't it be mixed? Is more correct? That's a good reason.
I guess mixed has uncompetitive. So there's a possibility that uncompetitive is doing it, but a holistic point of view is just uncompetitive like entirely.
Correct. So mixed has a mixture of competitive and non-competitive. So that's why you probably see a decreasing and an increasing with Vmax or Km. I have B as the choice, so, but it's still a good question if it could be mixed. But based definition specifically, we know that uncompetitive is always decreasing with both. So that's why it's a more correct answer, like the gentleman in the back said. Yes, so if you had maybe another value where you saw Km increasing as well, then you could say it was mixed. But because only Km is decreasing, we can correlate to saying that it's uncompetitive. Do we want to go over graphically what each one looks like? Or are we okay with that? No? Everyone's good? Okay. Moving on. The heme group in myoglobin binds the O2, to oxygen, which is the function of the polypeptide. All of the above? Yes? Awesome. So the polypeptide provides stability for the heme group, which is very nonpolar. That is true. B, the polypeptide prevents permanent oxidation of iron 2, which is also true. Now, what part of the polypeptide coordinates with iron 2? I'm sorry? The porphyrin ring is the actual, so that's a good guess, but that's not what's coordinating directly. Histidine, which histidine residue coordinates with the iron, and which histidine makes a hydrogen bond with the oxygen. So for C, what do we think? Because they're both histidine groups. I have, so you said E7 for which one? So E7, we have binding to the oxygen, and F8 binding to the iron. So I remember iron has an F, F8. Easy peasy lemon squeezy. That's horrible, I need to stop. All right, let's just continue on. All right. So the arrangement of the T state hemoglobin to the R state. D, B, a delta, D for delta. That's awesome, because that is correct. So what is happening with the movement with when iron is coordinated? Awesome, so it moves in line with it. What is the angle that it rotates in order to move in line? 15 degrees. Awesome. So that 15-degree rotation causes the iron to affinity bind. Any questions? Okay. 34. Which of the following increases the affinity of hemoglobin for oxygen? Increasing the affinity for oxygen. I hear C, I hear D. C. Why wouldn't it be D? So I always like to draw a little curve. So if we have a shift in the right and a shift in the left, what causes a shift in the left? What happens to pH in a shift in the left? Increasing or decreasing? Increasing? Shift to the left. Decrease? So pH is getting lower. Yes? So pH gets lower. So a shift to the right has pH going up. Do we all agree? So what about increasing in BPG? Which way are we shifting? To the left? How about increasing in CO2 concentration? To the left? Do we all agree? Well, I'm pretty sure pH to the right actually decreases pH. So it's not to the left. So this is decreasing in pH and this is increasing in pH. So as BPG binds, we have less, we can't bind oxygen more affinity. Like the affinity decreases. So when pH goes down, our oxygen affinity increases. Because my always example is when you're exercising, you want more oxygen and your pH in your body goes down because you're creating more hydrogen atoms to flow in the body and become released, which decreases your pH because a higher hydronium concentration means a lower pH. Does that make sense? So shifting in the right in the curve, we have our pH dropping and our CO2 concentration increasing. CO2, sorry, did I say something? So I think actually CO2 is increasing on the side. Yeah, so it'd be a shift to the left, sorry. So shift to the left with increasing CO2 levels. Does
Increasing CO2 levels decreases pH.
Yes. But corresponding, so you can breathe harder to release more CO2. So in a sense, getting rid of CO2 causes you to, I don't know. Don't worry about it. Because yeah, the Bohr effect doesn't talk about CO2 concentrations. I just threw it in there because I thought it was relevant. But I know BPG is relevant, pH, and temperature are the three things that we want to worry about. So shifting to the right, temperature goes which way? Temperature, does that shift the curve to the right or to the left when you're increasing? To the right. Any questions? I see some puzzled faces on.
Still
Are you saying the way you switch how the curve moves when these things happen?
Yeah.
Oh.
So the curve, so what's at the bottom of the curve? Oxygen. And what's on the y-axis? Saturation. So this is partial pressure of oxygen, and this is saturation of oxygen. So as that pH goes up, our saturation levels go down. So, or that's not true. So shifting it causes it takes a higher level of partial oxygen to saturate completely. So double check on this one. Just look on the PowerPoints and make sure that when there's a shift in the left, what's happening to BPG, what's happening to pH, and what's happening to temperature. We can come back to that one later on as well.
Well, think of it like this: like hemoglobin will release oxygen in the cells when CO2 is released because the pH drops. CO2 gets released and makes carbonic acid. And since that's more acidic, it'll release O2. So as pH goes down, affinity goes down. So your cells know where to drop oxygen off, where it's releasing more CO2.
Awesome. Okay, moving on. We can come back later on. So the classic experiment demonstrating the reduction and denaturing of RNA A could refold into the native form demonstrates blank. A. A primary structure can determine three tertiary structure. Yes? That is correct. So what type of molecules, or what type of reagents do we use to denature the molecule from a tertiary to a primary? Urea? What else do we use? Heat? Anything else? Detergents? So order-wise, what's the order that we want to use to take a molecule from primary structure to tertiary structure? So removing what creates the disulfide bonds back? Urea. So do we want to remove urea first, or do we want to remove the other compound?
Oh, they use the reducing agent also.
Reducing agent? What do we think? Because you would all agree that there's an order to it. Because if you take away one before, what happens is the molecule can't form to its tertiary structure. Detergent than urea? Do we all agree? So detergent. Okay, so we'll go back to that one as well. Moving on, what is the classification of alcohol dehydrogenase and an enzyme catalyzing the conversion of methanol to formaldehyde? Rhea first to reform this disulfide bridge, and then you remove detergent agents. I see. Thank you. So in the previous question, we're seeing that we want to remove urea and then oxidase. If we remove oxidase first and then urea, we have the inactive molecule. So remember, remove urea first, because no one likes to get peed on, and then oxidase. This is chapter 6, proteins, three-dimensional structures. Thank you. So for this one, it is a oxygen reductase. So forming the molecule, you have so in creating the double bond, what are we losing? Hydrogen. So what's something that usually takes a hydrogen? NAD to the NADH. Awesome. Any questions? Nope. Okay, moving on. 37. Which of the following is true regarding crystalline proteins? D? Which one? D, A, and B? So why do we think A? It's true, it's fact.
Because it's still in the same shape. So like if it was not in the crystal, it's not like we changed the shape of it.
Yeah,
That's the edge.
it's in that static form. So it's so a good idea to remember is like if you had to keep someone alive forever, you want to like chronologically freeze them. So like if you wanted to keep what is it? Oh, cryogenic. Yes. So they're not catalytically active. Well, they are in their form to be catalytically active. They wouldn't be able to probably do the reaction. So why is B true? So the diffreactive pattern observed during X-ray exposure to the crystal can be used to calculate the electron density map of the crystalline protein. So what do they shoot at this molecule, at this crystal? X-rays, because it's X-ray crystallography. And then what is the better resolution? Do we want a higher resolution or a lower resolution? Higher resolution? So we want that number to be bigger?
Well, you want your electron density to be smaller, but you like higher resolution.
You want higher resolution. So how do we get higher resolution? Smaller. What is it usually measured in? Angstroms. So that lower angstrom, so if it's over 1, that molecule starts to become more blurry and the waves and the pattern look more like mesh. But as that molecule gets that angstrom length gets smaller, we have a much more clear molecule. So D is the correct answer. And it wouldn't be C because the larger region in electron density would become, so we don't want a larger region, we want a smaller. Any questions? Okay, 38. Protein dynamics is a field of study that examines the movements within a protein. Which of the following type of protein structure determination would be the most useful to the study this type of change? Well, why wouldn't it be X-ray crystallography?
Can't move.
Yeah.
So it's static? We never learned about this, so we're never going to be tested. So that leaves this is a choice, and this is not a choice. So NMR is the correct answer. What type of NMR techniques can we use to use dynamics? Proton NMR? That's a good guess. Anything else? Nosy and cozy. So we have about 15 minutes left. Do we want to keep going through the rest of the questions, or do you want to go over your own questions that you have? Yeah, we can go through and see if we can fast fire the rest. So let's just do that and see what we get through. Okay. So an enzyme-linked immunoabsorbent assay requires D, antibody. Antibody that binds a protein of interest. So that's pretty straightforward. You just have to know that enzyme-linked immunoabsorbent assay is. What's another name for this? Lysa. Awesome. So 40, which of the following is required for effective catalyst of lysozyme? B. What do we think? She counted B and C. I have B being the correct answer. Because if the glutamic acid is a base, what does that mean about the charge? So the glutamic acid uses as a base at the end? Okay. Would it be, so it would be a weak base, if anything? Okay. I have B being the correct answer. Okay. So if it comes up on the test, if anything, I don't know if she'll argue that because it's hard to argue things once the final occurs. So I have B being the right answer. Just to clarify. Okay. C, or next one. In sickle cell anemia, which neutral amino acid residue replaces the negatively charged glutamic acid residue? What do we think? Oh, sorry. Here are the other choices. C. C is correct. So valine. So you could automatically go through and say what's not negative. Is glycine negative? No. Is adenosine even an amino acid? No. So that can't be right. And then that leaves tyrosine and lysine, which could be negatively charged amino acids. Or wait, no, lysine can't even be a charging. Can lysine be negative?
No.
No. So it leaves tyrosine and valine is the correct answer.
Question after neutral.
Oh yeah, so neutral, so glycine could be an answer, sorry. So that leaves tyrosine, could be neutral. These are polar molecules, adenozine's not even one. Any questions on that? Which of the following, all right, 42. What's our time? So which of the following characteristics of collagen structures listed below contribute to the tensile strength of collagen? I hear C, A. Why is it A? A is correct. I'm asking why. Why wouldn't it be due to the rigidity, due to the presence of glycines every 3 residues? So that glycine doesn't have what on it? So it has no functional group, so it's able to turn in the molecule. No. So that's not correct. If she hadn't put rigid, it's part of the molecule, but that's not why we have its strength to the molecule. So 43, which of the following amino acid groups would not make a good nucleophile catalyst? The methyl group? That's correct. So 44, during the course of the catalytic reaction, the following occurs. Substrate is covalently bound and oriented with proximity to the active site residues. Negative charge builds up on the substrate and is stabilized, and oxidation of the enzyme followed by reduction to the complete the catalytic cycle. What type of chemical species can facilitate these reactions? So I'm looking at catalytic reaction. So negative charge builds up and the substrate is stabilized. Oxidation followed by reduction in the catalyst. So I see negative and stabilized. What's something that we just talked about that has negative charges on it, but then we have a positive charge that stabilizes it? Magnesium. So transition metal ions, an example being magnesium. So I think that's all the time we have for now, just because there's only 5 minutes left in the session. So I will post the answers later on for the rest. And if you guys have any questions, you're more than free to welcome to come up and ask. And if you guys don't mind, before you leave, just turning in the surveys. Yeah, I can show you guys on the sheet. So we're on 43. So we have 45 being glycines, or gangliosides. Then the next one being D. 47 is C. 48 is A. 49 is D. 50 is C. 51 is C. 52 is C. 53 is B. 54 is D. 55 is A. And 56 is A, and 57 is A. 57 is A. Mm-hmm. Which one? 52 and on. We're sorry, we're kind of, yeah.